So I'd like to introduce our next speaker, Professor Ehud Cohen, also working at the Hebrew University of Jerusalem. And the title of his talk is The Orchestration of Proteostasis Across the Organisms. What have we learned from the worm?
Okay, first I would like to thank the organizers, specifically Karen, for putting together this meeting, and the friends from the UK, of course. And in the short time that I have today, what I'll try to do is to illuminate an emerging aspect of proteostasis, and this is actually the regulation of proteostasis across the organism by neurons. So what is proteostasis actually? As you all know, my lab and other labs have shown that aging regulates the integrity of the proteome, and it does it by regulating the ability of the proteostasis network to function properly. And what is the proteostasis network? So it's actually a general name for many mechanisms that act in concert in order to ensure the correct folding and maturation of nascent proteins, to make sure that proteins that fail to fold properly will be degraded by either autophagy or the proteasome. And as you all know, like in any other production process, efficiency is not 100%, and a subset of molecules fail to fold properly. They may undergo aggregation that can be deleterious. Aging affects different nodes of the proteostasis network, and we know that in general, this is a cumulative process of declining ability of the cell, and in this case, I'm going to talk about worms, to maintain the integrity of the proteome. So what other mechanisms that I didn't mention are part of the proteostasis network? And here I'm going to focus on three mechanisms which are known as the UPR mechanism, unfolded protein response. They are organelle specific within the cell. And today I'll focus on the heat shock response. And upon exposure to heat, proteins misfold, exposed hydrophobic domain tend to aggregate, and the cells have to respond to this accumulation of misfolded proteins. And the classical response is known as the heat shock response, involves the entry of the heat shock factor 1 into the nucleus, trimerization, and activation of gene expression, including of chaperones that help the cell cope with this stress. So what are the actually relations between the stress response mechanisms of the worms and the ability of the worm to cope with mild stress, which is caused by aggregative proteins? So historically, the heat shock response was supposed or thought to be regulated at the cell autonomous level. And this thought actually stemmed from the observations that cells in culture or yeast, when they are exposed to heat, they activate the heat shock response. However, a surprising study that was published in 2008 by the Morimoto laboratory have shown that in the C. elegans, in the intact organism, the heat shock response is regulated by neurons. And specifically, when Vina Pralad, at the time postdoc in Rick's lab, knocked down the activity of GCY8, which is critical for the functionality of AFD neurons, also known as thermosensory neurons, or of TTX3, which is responsible for some critical functions of the AIY neurons, the worm failed to activate the heat shock response in remote tissues. And this was the first evidence that actually in the intact organism, in this case C. elegans, neurons regulate the heat shock response of the soma. So two of my students at the time, Philippa Marks and Moria Maman, decided to screen for receptors in neuronal cells. At the time, we thought that we are screening for neurons at the AFD thermosensory neurons. You'll see in a few slides that this was not the case, that are critically important for this regulation or remote regulation of the heat shock response in other tissues. And what they did, they used RNA interference, fed the worms with a pre-selected set of neuronal receptor or RNAs that target neuronal receptors, and exposed the worms to heat and tested what the survival rates are. And as you can see here on the left side, DAF2 mutants, which are long-lived and highly resistant to heat. When we knock down this GPCR, which we called later GPCR thermal receptor 1 or GTR1, drastically reduced the survival rate of the worm, just like when we knock down the critical transcription factor DAF16. So we estimated that this receptor is expressed in the thermosensory neurons, because those are the neurons that are shown previously to regulate the heat shock response across the organism. But we found that this is not the case. You can see in the enlarged picture, I hope that you can see, that when we crossed worms that express GFP in the AFD neurons with animals that we created that express RFP in under the regulation of the GTR1 promoter, we saw that there is no colocalization. So we kept crossing worms until we found animals that show colocalization, and this was actually showed us that the GTR receptor is expressed in what's called chemosensory neurons, meaning that chemosensory are also thermosensory, and that the system that regulates the activation of the heat shock response in distance is actually much more complex than estimated previously. But how the ability to respond to heat is related to the ability of the worm to cope not with acute stress, but with mild stress of protein aggregation. So in order to address this, we used worms that we like to use in the lab. This is a strain which is called CL2006 for Chris Link 2006. This is the researcher who created them in 1995. And what Chris did at the time, he cloned a mini gene of A beta, as you all know, the short peptide which is associated with Alzheimer's disease in human, under the UNC54 promoter. This is the promoter of the heavy myosin chain. Thereby, it drives the expression of A beta in the worm muscle. And as a result, the worms become paralyzed, but they can still move their head. So if you look at the worm in the picture on the bottom right of the slide, you'll see that the worm consumed the food within the reach of its head because it actually cannot move. And we use this simple phenotype in order to measure proteotoxicity. And here we had a great surprise. So if you look at the green line, you'll see that these are animals that were treated with GTR1 RNAi, meaning that they cannot activate the heat shock response in the soma. However, the rate of paralysis was lower than control animals that are represented here by the blue line. And this means actually that the ability to respond to acute stress comes at the expense of the ability of the worm to maintain proteostasis upon mild stress. Similar results that neurons actually regulate the response of C. elegans to misfolded proteins was published about the same time by the Morimoto laboratory, and they show the same thing for worms that express polyQ, another aggregative protein which is associated with the development of at least 9 human disorders. So just like the heat shock response, proteostasis is regulated at the organismal level by neurons, and these are not only the thermosensory neurons. And the question that we ask next is whether the proteostasis network differentially responds to these similar proteotoxic challenges, or whether there is a common activity or common set of genes which would be activated upon stress of different types. And the rationale here was to search for components of the proteostasis network that were shown to protect worms from polyQ-mediated proteotoxicity that are deleterious in worms that express A beta. So we took advantage here on screens that were performed by other laboratories and defined subsets of genes that are critically important for protection from polyQ. What are the models that were used? So again, the Morimoto laboratory created subset of worms that express polyQ of different sizes, of different lengths. Up until 35, it's the protein has low tendency to form aggregates, but if you use worms 35 and more repeats of polyQ, they become aggregative, as you can see in this picture, and actually this aggregation leads to reduced motility. And how this motility is used, here the assay is a little bit different. Since the worm do not really become paralyzed within the time frame of the experiment, what we do, we use what is called the trashing assay. And the idea is based on the observation that if you place a worm in a drop of liquid, they tend to start kick, as you can see here in this essay. And the number of kicks per given time can also serve as a measure for the rate of proteotoxicity. So Hannah and Philippa, two talented students from the lab, decided to screen for such chaperones that can actually, if you knock them down, they aggravate the toxicity of poly-Q, but on the other hand, they protect the worm from A-beta proteotoxicity. And what you can see in here, that torsins, which are subsets of chaperones that were shown previously to be critically important for the maintenance of proteostasis in poly-Q expressing worms, actually their knockdown protected worms from A-beta. And when we looked at worms that express the two aggregative proteins in neurons or in muscles, you can see here the example of neurons, we got exactly the same thing. Regardless of the tissue of expression, the knockdown of torsin aggravates toxicity of poly-Q, but protects the worms from proteotoxicity. And obviously the next question was, what's the mechanism? How does it work? So in order to address this, we've done several things. I'm just going to focus on one experiment. And here what we did, we used RNA-seq in order to test whether we can find genes that actually respond in opposition to the knockdown of torsins in worms expressing poly-Q or A-beta. And what I asked the bioinformatician is to generate a list of genes that respond in opposition. And as you can see here on the left side, the bottom four genes are actually coding for neuropeptides. And in last year, in 2020, the Dillon lab found that neuropeptides actually are important as regulators for ER stress. So it seems to us very logical that neuropeptides would be involved here. But obviously, we tested it. And as you can see in here, on the left upper panel, you can see that when we knock down those neuropeptides, one at a time, we got protection from A-beta. On the right side, you can see that motility of poly-Q worms was reduced. So there's neuropeptides actually. When we knocked them down, we got the same opposing effect which we observed when we knocked down torsins. Similar effects were obtained when we knocked down all four neuropeptides in parallel. So which neurons are involved in the secretion of these neuropeptides? So let's see if it works here. No, it's a movie. You can see that we took those worms, we expressed the neuropeptides, the GFP under the neuropeptide promoter, and we could actually recognize the neurons that secrete those neuropeptides. And just to sum up, because of the sake of time, we found several sets of neurons which are important for those opposing effects. And basically, the conclusion here was that somehow neurons get the information from the soma, informing them which aggregative protein is challenging the somatic cells, in this case, muscle cells. And upon receiving this information, the neurons of the worms formulate a secretion of different sets of neuropeptides in order to activate transcription factors. I don't have time to go into the data, but we know that this is SKIN1 or NRF, the ortholog of the human NRF in the worm, in order to specifically activate subset of genes that provide the worm protection from the specific type of challenge that the animal faces. But one more thing that I want to highlight before my time is up is that some of those neurons, and you can see on the left side that we labeled ASI and NSM neuron, those are serotonergic neurons. So whether serotonin is also involved in here? So Vina Prahlad, now an independent PI, published a paper in 2015 showing that neuronal serotonin release triggers the heat shock response in C. elegans, even in the absence of temperature increase, and it was actually recapitulated by other labs and that showed that the serotonin release is important for the maintenance of proteostasis. But how about mammals? So another paper, which was published in the same year by Patricia Messiel, shown that serotonergic signaling suppresses ataxin 3 aggregation and neurotoxicity in mice. And actually, the lab of Patricia and myself, situated in Portugal, they are interested in Machado-Joseph disease, and ataxin-3 is the poly-Q containing protein that its aggregation underlies the development of the disease. And they have Machado-Joseph disease mice, and treating the mice with SSRIs that prevent or actually reduce the uptake of serotonin protected the animals from disease. So SSRIs as novel drugs for proteotoxicity, there are several indications that they could be served as proteostasis promoting drugs. I'm just showing you one example that was published a few years ago. And in this study, it was shown that in worms, paralysis of the same model that we use, CL2006, can be mitigated by fluoxetine or, as you probably know it, Prozac. So, in conclusion, what have I shown you today? First, that the ability to respond to heat sometimes comes at the expense of proteostasis maintenance. This was very surprising. Neurons regulate proteostasis across the organism by the secretion of signaling molecules such as neurotransmitters and neuropeptides. Maybe neuropeptides and or neurotransmitters modulators, such as SSRI, should be considered as components of future therapeutic cocktails for the treatment of neurodegenerative disorders, and whether and how intertissue communication regulates the aging-associated proteostasis in humans is obviously a question that should be addressed. At this point, I would like to thank members of my lab, specifically Hannah, Philippa, and Moria, who did the work, our funding agencies over the years, and thank you for your attention.
Great, thank you. So the floor is open for any questions.
Are there any studies done cross-sectionally on humans to show if some people that took Prozac versus others are more protected for aggregation in the brain? And are there any other drugs that might affect on the peptides that you identified, which are non-serotonin dependent?
So those are two great questions. So I searched the literature, and to the best of my knowledge, no such study was done in humans, although maybe I missed something. Yes, I definitely believe that in the future we'll have to stop thinking of neurodegenerative disorders as one type of disease. We all know that Alzheimer's, for instance, is a very complex syndrome that has many manifestations. And I think that in the future we'll have to better characterize subtypes of patients and formulate cocktails. Now, what could be in such cocktails? Could be specific neuropeptides, could be anti-inflammatory or other drugs that are currently in the market or will enter the market. And I think that we'll have eventually to think in a combinatorial manner. Yeah.
So, great talk. Thank you very much. Actually, depression is a risk factor for Alzheimer's disease, and I didn't really think about the mechanism, but you actually gave perhaps a mechanism, so this is very interesting.
It is possible, but I must stay humble. You know, it's very difficult to measure depression in worms, so I don't know whether it's relevant, but it could be. It definitely could be.
Thanks.
Yeah.
Thank you. Are there any more questions?
Okay. Thank you. Thank you, Mellissa.
Thanks.